Good morning. Biology 3260. Say hello to our audience members. Good morning.
Hi.
We started off this first unit of this class talking about how there were a lot of ways that cells are really similar to each other. And one of the ways that we said cells were similar to each other is up on the board now. And it said that cells are in community, they live in tissues, and they actively communicate with other cells. So how do cells respond to a signal? Well, they can do several things. And I just illustrated this with the BMP signaling. They can tell the cell that it should survive. They can tell the cell that it should proliferate, that is, to grow and divide. They can tell the cell that it should differentiate if it is a precursor cell into a specific fate. And they can also induce the cell to die. So how does it do that? Well, here's a big overview of what we're going to talk about for the rest of the day. You're going to have some kind of a signaling molecule. We'll talk about what those are. That's going to bind to some kind of a cell surface receptor protein. We're going to talk about what those are. And that is going to induce the receiving cell to do something. It could be a very fast response, like altering protein function. Or it could be a relatively slow response, something that takes minutes to hours, that actually changes protein synthesis in the cell. End result of any of that is that the cell that's responding is now doing a behavior that it didn't do before. And you'll remember a couple of weeks ago when we were talking about cell structure, I divided you into two cells. This is one cell, and this is the second cell. And then I asked for everyone who is part of the plasma membrane of your cell, please stand up. So plasma membranes, what kind of importance, or what's maybe one of the most important organelles are inside of one of our cells? Nucleus. Yeah, good. Thank you. That's what I think the most important one is too. Can I have two brave volunteers that are not part of our plasma membrane in each cell to stand up? Hopefully you're sitting close to each other. You're really plasma membrane adjacent. Okay, we'll make this work. All right, so nuclei, raise your hands for me so I know where you are. Okay, good. Is the nucleus, everybody, are our nuclei easy to get to? No, why? Right, it's surrounded by the plasma membrane, so it's kind of protected, right? And do we want the nucleus to be protected? Why? It has the DNA in it, right? So I don't necessarily want all kinds of things being able to access my DNA, because then bad things happen. But I also need for cells to be able to access the DNA if I need to make a response as a cell, right? To a signal to my nuclei, we can make it travel pretty easily. It's small, it's hydrophobic, it can get through. Right. Now, what if our signal is a big fat protein? I even made it pink today so you could see it. So what happens if it goes to the plasma membrane? It gets blocked. Why?
It's too big.
It's too big to get there, right? So the plasma membrane is effectively doing its job. It's keeping things that are too big out. So, is there a good reason to have a signal go from a signaling cell, let's say that you guys are sending the signal, and you guys are receiving the signal? How might that work? Let's say you need to get it back over here.
So, I can pass it along the membrane.
Okay. You can also come up with your own ideas here too. It hasn't gotten inside the cell yet. How might I be able to get it inside the cell? What if, Cesar, help me out. What if that particular piece of the plasma membrane was actually a protein? that had an arm sticking out and an arm sticking inside. So now we're not just talking about one cell sending a signal to another cell. We're talking about a coordinated activity has to happen in this cell, and an equally coordinated activity has to happen in this cell. All right, so how does a cell respond to a signal? And now, instead of thinking about what does the cell do, I want to know how does the cell elicit that response? So molecularly, what does the cell do to respond? So now we've gone from our example, we've put some names to all of the different players in this cell signaling pathway, but we haven't really given them specific molecular identities yet. These are really very generic terms. But the term ligand and the term receptor are two important words that you should remember. So the ligand is synonymous with signal. Okay, that's the signal that's being sent. And the receptor is the biomolecule that receives that signal. If you wanted to then put what we just talked about into some more relationships between one cell to the other, it might look a little bit like this. So here's your signaling cell up at the top corner. And the signal itself, the ligand, is in these little red circles. The signaling cell is trying to send a signal to this responding cell, which is the one at the bottom here. This is the responding cell, signaling cell up at the top. So the signaling cell is going to produce these little ligands, these little red circles, and then release them into the extracellular space. Those ligands can then be received by a specific adapter, which turns that receptor on, or activates that receptor. Now remember, the important part is that the receptor here, if it is in the plasma membrane, the receptor never moves. Okay? In this case, what the receptor does is it causes some activation of some adapter protein inside the cell. That will then trigger a cascade that binds to another one, that binds to another one, that can then ultimately affect this effector protein. That effector protein might have the ability to actually move into the nucleus, and there are several different classes of proteins that are able to do this, usually guided by another protein. The end result of that is changing something, and most of the time, when we're talking about this in developmental context, many of the times, it's changing gene expression. You fed 40 flies, 40 females in 2 different vials. You may or may not have all 40 of them still left, depending on how many you let go when you fed them yesterday. So our goal today is to dissect cleanly 10 ovaries, 10 pairs of ovaries, I should say. That gives you at least 10 to mess up, at least 10 to maybe get partially, but to clean them up a little bit, and then assuming that you lost 10 of them yesterday when you tried to feed them, okay? So there's no pressure today. The whole goal is to do this as a practice, okay? So again, I want you to experiment a little bit today with like what feels best for you, what can you see the best on your microscope with your eyeballs, right?
I would agree.
Okay. You see it? What do you think like might be nice? So you've got one hand over, one hand over. You might want to put some more liquid actually in your dish.
Yeah, I
It looks
think
like
the
you're in
raw
a
is probably
tiny
one
little bit of liquid. So let's try.
Also, I would probably give up on that.
Well,
Personally.
she's got it. You got one. Yeah, I see it.
It's not a big deal if you have a test.
No, it's not a big deal if they're not attached.
It's more satisfying
And,
though. when
yeah,
they're
it is more
attached.
satisfying when they say
It's so
attached.
satisfying.
But in the long run, no, it does not happen. I see them. Yeah, perfect. So what I do is dissect because you've also got flies that are under anesthesia, right? And the longer you leave your flies on anesthesia, you will kill them eventually. That would be bad. So my suggestion, the way that I do it is I try to get all of the flies dissected as fast as I can, and then I go back and tease them apart later when I can turn the gas off. Just for, safety. So, yeah, your scopes go from like 12 to 30 something. I usually have mine at like 25. I zoom way in on her. You're also doing the overhanded part too. After you try this fly, try holding on to your both forceps as if you would hold on to a pencil in both hands.
Like have it resting on your ear like this.
You should try that one the way you have it right now. Yeah, it's
Okay,
okay.
me too.
Stick it down in the liquid then.
You're good.
Then with the hand that's not holding on to her. So I think that's the other reason that we were talking about whether, like the girls were saying that they dissect above the liquid. To me, that's really hard because then you have to keep it above the liquid. So I take the forceps with my fly. My fly is in my forceps, and I plunk them down on the bottom of the dish. So it's anchored up against the bottom of the dish. And then I put my the focal plane of my microscope and the magnification so that it's always down on the bottom of the dish. And that way it's anchored better. And then I can pull things from there. So basically, like, your non-dominant hand is really like, it's your anchor. So you're pushing the fly down against the bottom of the dish, and you're holding her still while the other hand is doing all of the work.
I pulled them out all at once, but like the back is still attached.
Then you still have to get the back off. Yeah, no exoskeleton.
Also,
It'll
it
just
helps
ruin your microscope.
if you use like the needle. Like if you rip everything, you know what I'm saying?
Oh, to go back and clean it up with the needle.
I
Yeah.
just really that's not how you
Yeah,
like
it's so sad.
too. So I'm like freaking tearing their heads off left and right. It's so irritating. And then a lot of times I'll have like skin stuck on
It's
like
like
their ovary.
the same
and I have to
thing.
Yeah, it is.
Extremely small.
Yeah, it is. And the only real difference between them is the way that the tissue is organized. So like in human ovaries, there's no real logical like progression to it.
Yeah,
They're
in a clump.
yeah, and they're kind of where the primordial follicles are kind of more towards the cortex a little bit. Or I guess those are more interior, and then the ones ready to rupture are more on the outside. But there's not really like it happens all the way around the organ. Whereas these, it's literally a line.
See the progression.
Yeah, exactly. And so when you're thinking about, how do you sell this as a research model, like that's really important because that means if something is not happening developmentally, you see it immediately.